Okay, so today we're going to go over the Kreb cycle. That'll be fairly quick, and then we will go over the electron transport chain, which pretty big process. So anyway, so we already went through glycolysis. In glycolysis, happened in the cytoplasm. We took glucose, we broke it down, did some intermediate things, which we talked about in that video, but our end product was pyruvate. After this, as long as oxygen is present, we went through pyruvate oxidation, where we are going to take that pyruvate and we are going to make it into acetyl coenzyme A. So we went from pyruvate, which was a 3-carbon molecule, into acetyl coenzyme A, which is a 2-carbon molecule. And in that process, with losing that carbon, we gave off carbon dioxide, which is where that 1 carbon went. So we are going to do the citric acid cycle, or the Kreb cycle, today. And from pyruvate oxidation, our product, which was acetyl coenzyme A, we are going to take the coenzyme A, which 2 carbons, all right, and we are going to drop it down into our citric acid cycle, or you could say our Kreb cycle, either one works. Now, it's a cycle, okay? So it's just going to keep repeating itself as long as we have acetyl coenzyme A fueling it. Now, before we can drop acetyl coenzyme A into the Kreb cycle, we have to have something formed by the Kreb cycle for it to combine to. Now, that is going to be oxaloacetate, okay? And it has 4 carbons. So what we're going to do, draw my arrow a little differently here. We are going to take our oxaloacetate and we are going to combine it with our coenzyme A here. And we are going to form, now remember, we have 4 carbons from our oxaloacetate, and we have 2 carbons up here. We are going to form citrate. You can also call it citric acid. Now, 4 plus 2, it's going to have 6 carbons to it. All right. So, going on from there, we're going to take our citrate. All right, we're going to bring, in each one of these things, guys, just like glycolysis, we are going to have some enzymes. We're not going to worry about them too much. I'm worrying about you knowing how the products are formed, okay? And your book gives a big diagram, but it has all the chemical structures of them, and they can get a little intimidating. So anyway, enzyme here, we're going to use isocitrate dehydrogenase, okay? Dehydrogenase. It's going to have something to do with water, so we're going to add some water into here too. So this is why we need water, drinking water. All right. Now, in this process, 2 things are going to happen before we get our product. We're going to take NAD plus, we're going to give it an electron to make it into NADH, and we're also going to release, let me write release there for you guys. We're going to release carbon dioxide. Now, the NAD plus, okay, we gave it an electron, cool, not too big of a deal there. But the second part, we have citrate, and we took away carbon dioxide. I'm not worried about the oxygen there. I'm worried about the carbon. So in that, we are going to have something different now because we got rid of a carbon. This is called alpha ketoglutarate. Okay. Do you got to know the fancy names? No, you do not. Do you have to know that we go from a 5-carbon from a 6-carbon molecule to a 5-carbon molecule? Yes, you do. All right. So we now have a 5-carbon molecule. Okay. Once again, going to the next one. So every time we take away a carbon, we have an electron that we can use. So once again, we are going to take away another carbon. We're going to have NAD plus going to NADH, and we're also going to release carbon dioxide. Okay. In doing so, we now have 4 carbons left over. So we're going to call this succinyl. Fun name. Coenzyme A. Big thing that you have to know here. It has 5 carbons. Okay. We are now going to transform our succinyl coenzyme A. All right, and we're just going to make some slight modifications to it. Nothing is changing with our number of carbons. So we are going to transform this into succinate. In doing this, we're going to take GDP, which if you remember, it's guanine diphosphate. We're going to transfer that into GTP. If you remember, we used this whenever we went over our primary or first and secondary messengers. Okay. And also, out of this, we're going to take ADP, transform it into ATP. So we get ATP out of this, we get GTP out of this. 2 different things we can use for energy. So now we have succinate. There's a bunch of different intermediates here, guys. I'm going to draw them in black because not too much happens here. We're going to have fermate. Fermate there. That's going to be transformed into malate. Okay, and you can see, I mean, succinate is still 4 carbons, and the goal was to get to oxaloacetate, which is 4. So we're not doing anything else with the carbons here. But some different things are formed in these transformations. Here we're going to get NAD plus transforming into NADH. Okay, so NADH, a good electron carrier for us. And then down here, when we go from succinate to fermate, we're going to take FAD and we're going to get FADH2 out of it. Okay, so this is our Kelvin cycle. Okay, all together for products. Let me put this in a different color here. Go with a nice pink. I don't know. All right. So for products, guys, yeah, you can count them. Look, NADH, there's 1, there's 2, there's 3. FADH2, there's 1. GTP, there's 1. ATP, there's 1. Okay? And we release 2 carbon dioxides. But products, don't forget, guys, we have 2 acetyl coenzyme A's. So we have 2 terms on our Krebs cycle. So for products, we are actually going to get 6 NADH. We're going to get 2 FADH2s. I'm going to get 2 ATPs. Going to get 2 GTPs. And then lastly, we're going to release a total of 4 CO2s. Okay? But just so you know, that is released. All right. So there is your Krebs cycle or citric acid cycle. All right, if you guys want to take a screenshot of that. I tried to make it as simple as possible for you. Okay. That ball trick does not work, guys. I'm trying to keep the lights on. Okay. So there is, like I said, guys, Krebs cycle. All right. So here is the book versions of it. All right. You guys can see they give you all the intermediates. They give you all the chemical structures. We're not going to worry about all those. Okay. All right. So this is everything we went over, guys. I don't need to go over all this again, all the different steps. I will mention that NADH and FADH2 are later going to be used by the electron transport chain. So if you remember, in glycolysis, we formed some NADH. So that's only from glycolysis that we get some of our NADH. We don't get any FADH2 from glycolysis, okay? So glycolysis is only producing NADH that we're going to use in the electron transport chain. Now, the Kreb cycle, that gives us both. That gives us our NADH and our FADH2. So we can combine the NADH from the Kreb cycle with the NADH from glycolysis and use both of those in our electron transport chain, okay? All right. So after this, we're going to have, it's called oxidative phosphorylation, also known as the electron transport chain. And this is where we are going to make most of our ATP, okay? So at the beginning, guys, we started in the cytoplasm with glycolysis. We then moved to the mitochondrial matrix, which was the inside, way inside of mitochondria to do the Kreb cycle. Now we're actually going to move outward and go to the inner membrane of the mitochondria. And the cristae, if you remember, the cristae are all the folds. What that does is it increases the surface area, and I will show you what is so important about that increased surface area when we go over the picture. But just so you guys know, the big thing is the inner membrane. It's going to be folded, okay? So we have those cristae, so we increase the surface area. That makes more room for more oxidative phosphorylation, more ATP gets made in that. Okay, so here is the last part, guys, and then there, if we look here, there's our cristae, okay? We're going to be moving from the matrix to the area in between the outer and inner membrane and back in and back out, okay? And you'll see in the diagram that we're going to go over. Okay. So here is the book's diagram of the electron transport chain. I'm going to do it a little bit differently than the book. I like my diagram a little bit better, okay? So what we're going to do is we are going to pass our electrons. So the NADH and the FADH2 are going to pass these electrons through different proteins, and then there's also these things called cytochromes, okay? They're like proteins, but they're not going to give us as many protons, okay? So, all right. Okay, so we already went over the first one, mitochondrial matrix to that inner membrane space. That's the area in between the outer membrane and the inner membrane of the mitochondria, okay? What we're going to do is we are going to make a gradient of our H pluses, of our protons. And this is going to allow us to take ADP and convert it into ATP. And this is called chemiosmosis, okay? The end of our electron transport chain is going to be this guy here. This is called ATP synthase. It is almost like a machine in your bodies, in each of your mitochondria, that is actually going to generate some kinetic energy. So if it's not moving, it has potential energy. If it's moving, energy of motion, that's going to be kinetic energy. So what it's going to do is it's going to start to spin, and as it spins, it creates kinetic energy, and that kinetic energy allows ADP to get converted into ATP. Okay. We're going to skip this guy here. So here is my diagram, guys. This is one I pre-drew before we started. I know it's not very pretty. I apologize, but it'll do the job. So we have our inner membrane right there. Okay. And I'm not going to keep that on there. So we have our inner membrane right there. Now, with that inner membrane, we're going to, like I said, transfer here, transfer back over. We're going to keep going back and forth from the inside to the outside. All right. Now, what's going to happen, guys, is we are going to have some proteins, all right, and we are going to have some cytochromes, as we talked about. I'm going to label our proteins here. This is going to be protein number 1 in red. The orange one is going to be protein number 2. All right. Next one, this guy here, the light blue one, protein number 3. Pink one is going to be protein number 4. Okay. We're going to have our cytochromes as well. This one here, okay, this is called coenzyme Q. And the gray one here, this is going to be cytochrome C. All right, so let's begin. Right now in our matrix, we are going to have a bunch of protons, and they're all just going to be chilling there. Whole bunch of protons. I'm drawing them all in this one area. They will be all throughout the inside of the mitochondrial matrix, but if I draw them all throughout, it's going to make everything extremely packed. So I do not want to do that. All right. So what we're going to do is we are going to take our NADHs. All right. So the first thing we're going to do is take NADH. And where do we get this NADH from? Well, we're going to get it from the Kreb cycle. We're going to get it from the electron transport chain, or I'm sorry, from the Kreb cycle and from glycolysis. And we are going to allow it to lose an electron and transform it into NAD+. Okay? Now, once that electron is lost, that electron is going to go right here to number 1. Here's our electron. That electron goes to number 1, and this channel is initially closed. Whenever that channel opens, it's going to allow one of our H's to go through and to the other side. Okay, now let's go back to the chapter on diffusion and osmosis. We have a whole bunch of H's in the middle here. We have very little on the outside. Since we have very little on the outside, this will happen naturally. We will not need ATP. We will not need to expel energy or use energy for this to happen. So we're going to take our electron, it's going to open this up, and then it's going to let the H's through. Now, originally, if we're talking about our protein number 1 here, all right, I'm going to say this is our change of energy. We are going to have very little energy, but when it gets that electron, the energy shoots up for protein number 1, okay? It does not want to have a lot of energy, so it's letting that H through by the electron. So what it's going to want to do is it's going to want to get back down with that energy, okay? It wants to get rid of that energy. In other words, it wants to get rid of that electron, okay? So what it's going to do is it's going to pass that electron on to our second player here, okay, which is coenzyme Q. So same thing happens here. The electron is now at coenzyme Q. All right. We do not want all that to happen. We don't want it to have that electron because the same thing happens. Energy goes up. So it's going to get rid of that electron. And we'll talk about what happens with that electron in a minute. So protein number 1, this is going to be our NADH protein. Protein number 2, and we'll come back to coenzyme Q, and I'll explain to you guys what that does. Protein number 2 is going to take our FADH2 from the Kreb cycle, and it's going to make, it's going to make it into FAD. Okay. And then what's going to happen to the FAD and the NAD+? The NAD+ can go back to glycolysis or the Kreb cycle and turn back into NADH and then go back, obviously here. The FAD is going to go back to the Kreb cycle only because glycolysis doesn't produce any FAD. And it's going to get transformed back into NADH2. Okay. Now, in this transformation, guys, we are going to get some protons broken off. So this is going to make some protons. It's also going to make some electrons. So I'm going to draw that electron in orange just to keep it consistent with that protein. Okay. This protein number 2, again, same thing. It gets that electron. It's a high energy electron. Oh no, it doesn't want that high energy, all that high energy. So what does it do? It transfers that electron to coenzyme Q. Now coenzyme Q has more than one electron. Okay. It's going to get some electrons, 2 from FADH2. It's going to get an electron from our NAD plus. So the thing about protein number 2, okay, which deals with FADH2, is if you notice it does not have a passageway. Okay, it doesn't have a passageway through it. So we're not going to get any H's into that area in between the outer and inner membrane of the mitochondria there. But here's what's going to happen. Coenzyme Q is now, and I'm going to draw these in green, is now going to take these electrons, this one and this one that went from here, and it's going to take those electrons and transfer them over to protein number 3. In transferring those electrons to protein number 3, if you notice protein number 3 does have a passageway, it's going to let some protons into that area out there. Okay? Same thing happens here. It has those electrons. It doesn't want the electrons. It's going to transfer them to cytochrome C. Cytochrome C, no passageway there. Cytochrome C is going to take those electrons, pass them on to protein number 4, open that channel up. That is going to allow more of these electrons to come through. All right, they'll go up here. I apologize, I'm going to make more stuff here. That's why I don't want to connect that line there. But more are going to go through. All right. So we're going to then take all these protons that are out there. So we have all these protons. I'm just going to draw a bunch of pluses over here. Okay. The protons are now building up in that area outside of the matrix. Okay. So our lovely lights went on again. So what we're going to do, guys, is we are going to pass these protons back into the mitochondrial matrix. Okay, so they're going to pass through, and they're going to go back inside through this area here. Now, what is this guy? Well, we talked about him earlier on a previous slide. This is ATP synthase. ATP synthase has this propeller, I guess you can call it, on the end, and that's going to generate our kinetic energy. Okay, so what we're going to do here, guys, we're going to pass our protons in. Now we have all these H pluses in here. They're coming back through from the outside. Okay, they're these guys, right? And what we're going to do is when they come through, they're going to get this propeller working. And what that propeller is going to do is going to take our ADP plus a PI, phosphate, and it's going to transform it into our goal, which is ATP. Okay? And that is what we are trying to make. Now, the one thing that we did not go over yet is throughout this whole process, we said glycolysis occurs without the presence of oxygen. It's anaerobic. Okay? Then we talked about if oxygen is present, we'll go through the Krebs cycle, we'll go through the electron transport chain, but we never talked about why we need the oxygen. Okay? This is where the end comes in. So, we are going to take these guys here, our protons, we are going to combine them with oxygen. And out of that, we will get water as a product. Okay? So, we have our ATP synthase. The protons that we moved outside, they're going to come back through. Those protons, sorry, I think I said proteins. Those protons, they come back through. They are going to generate ATP synthase to spin, creating kinetic energy. The ADP is going to combine with a phosphate. They're going to stick to the propellers here, okay? They're going to get some energy. That energy will allow the ADP to combine with the phosphate to form ATP, okay? And that is our electron transport chain. Now, how many ATPs are we going to get out of it? Well, let's do our totals here. Okay. In total, we had 10 NADHs. Okay. If we look at these NADHs, let's talk about the electrons. The electrons went through, I'm going to start with number 1 here. We went through 1. Nothing happened there. Okay. It coenzyme Q. Nothing happened. They then went to number 3. Oh, look, it let a proton through. Okay. So that's the second one. They went over to number 4. They let a proton in there. So they did a total of 3 proteins they touched. Okay? That equals, in the end, we will get 30 ATPs out of that. Our other one, we had a total of 2 FADH2. How many proteins did they touch? Well, if you remember, we didn't worry about number 1. FADH2 started at number 2. Didn't let anything through there. It went to number 3. Yeah, it let stuff through there. Number 4. Yeah, it let stuff through there. So it touched 2 proteins and allowed protons to move through 2 proteins. So 2 times 2 is 4 ATP. In total, we get about 34 ATP from the electron transport chain. Add that to our 2 from glycolysis and our 2 from the Kreb cycle, and we get a total of 38 ATPs per 1 glucose. And that is our goal of cellular respiration. Okay, so for every molecule of glucose, we're able to go through glycolysis, the Kreb cycle, and the electron transport chain to get 38 ATPs. Okay. All right. That was a lot. Here's the book thing, guys. We're going to skip all over that. Your book says it's about 32 ATPs. Yeah, I just showed you it was 34. Okay. So it's different, depending on the book. But yeah, I'm going to go with 34 there. Okay. Also, your book says that, in the end, we might get 26 or 28, depending on, all right. If we generate the correct amount of NADHs and the FADH2s, should be a little higher than that. All right. So we are good there. All right, guys. Next time I will do a short video on fermentation. So we will call it a day, and I'll see you next time.